Okay, so welcome back to this next video in which we are discussing Alzheimer's disease and Alzheimer's disease medications. Okay, so two little points that I want to raise from the previous video. Firstly is the correct spelling of annihilation. This is not the correct spelling of annihilation. Okay, so the correct spelling of annihilation is double N, okay, so Annie, and then halation. Okay, so it's not too bad after all. Annihilation. Right, the other thing I want to raise is something about hyperphosphorylated tau. In my pictures, I have suggested that hyperphosphorylated tau always has 3 phosphate groups on it. That was not intentional. I did not specifically say that hyperphosphorylated tau had 3 phosphate groups on. In reality, hyperphosphorylated tau has significantly more than 3 phosphate groups on, and each tau is likely to vary in exactly how many phosphate groups it has on it. Okay, this was just to illustrate the point that they've had a lot of phosphate groups added onto them. So don't read into that more than there is, basically. Don't assume that it is actually that each one gets 3 added on. They get far more than that added on, and different tau proteins will end up with slightly different numbers added on. Okay, right. So, what we now want to discuss then is the Alzheimer's disease medications. Okay, now the first thing to say is despite significant progress in the understanding of the neurodegenerative process underlying Alzheimer's disease, so despite significant progress in understanding the formation of the amyloid plaques and the neurofibrillary tangles, we have no drugs that are currently used that stop the formation of amyloid plaques or neurofibrillary tangles. Okay, instead, the 2 types of drugs that are used for Alzheimer's disease are merely treating the symptoms. Okay, one is acting slightly earlier on than the other. Okay, actually it's unfair to say that Mementine is a symptomatic treatment. One class of drugs, the first class of drugs that we're going to look at, merely treats the symptoms. Mementine seems to slow down the neurodegeneration. It doesn't stop the formation of the amyloid plaques or the neurofibrillary tangles, but it seems to reduce the abilities of those things to actually result in dead neurons. Okay, so we're going to firstly start off with this class of drugs that is going to be cognitive enhancers. Okay, so they're going to treat a symptom of Alzheimer's disease, and then we'll move on to looking at Mementine, okay, which will give us some insight into how maybe the amyloid plaques and the neurofibrillary tangles actually result in neuronal death. Okay, right. So then, the first category of Alzheimer's disease medications then, okay, this is the category of acetylcholinesterase inhibitors. Okay, so remember earlier on when we were discussing which portions of the brain are particularly sensitive to Alzheimer's disease, I told you about the basal forebrain nuclei, okay, which were cholinergic nuclei, and I said that the loss of cognitive function in early Alzheimer's disease wasn't thought to be because the cerebral cortex had degenerated yet. That's what happens in late Alzheimer's disease. But in early Alzheimer's disease, the cognitive decline is believed to be because the cholinergic neurons in the basal forebrain nuclei, for instance the nucleus basalis of Meynert, have degenerated very early on. They're very, very sensitive to Alzheimer's disease pathological processes. Okay, and the loss of those cholinergic neurons means that acetylcholine being released into the cerebral cortex goes down, and acetylcholine has this regulatory function. It regulates how active the cortex is. It regulates your cognitive ability. Okay, so usually when we're feeling tired at the end of the day, our cholinergic transmission in the cerebral cortex will be down, and that's why our cognitive ability is down. So in Alzheimer's disease, if these neurons degenerate, your acetylcholine in your cortex is permanently down, and that means that your cognitive function is permanently down. Okay, so we could treat this early cognitive decline in Alzheimer's disease by increasing cholinergic neurotransmission in the cortex, and that is the basis of the use of the acetylcholinesterase inhibitors in Alzheimer's disease. Okay, right. So, in order to discuss how these drugs are going to work, we need to just be aware of how cholinergic synapses work. Okay, so let's say this is the axon terminal of one of these cholinergic neurons, which will have its cell body in the nucleus basalis of Maynard. Okay, so here is our axon terminal. Okay, now when this neuron fires an action potential, the action potential will propagate along the axon all the way to the axon terminal here, which is far, far away in the cerebral cortex. Okay, and I'll colour in the axon terminal here in turquoise. The axon terminal will then release the neurotransmitter acetylcholine, okay, onto the postsynaptic neuron. Okay, and here's a dendritic spine of the postsynaptic neuron. Okay, so this is our postsynaptic neuron. And the acetylcholine will then act on acetylcholine receptors on the postsynaptic neuron, which might be ligand-gated ion channels or they might be muscarinic receptors. Okay, right. Now, how then does the acetylcholine get removed from the synaptic cleft? Okay, because the acetylcholine can't be left there forever, otherwise it would continue signaling on and on and on. After the neuron has ended its action potential, you want the acetylcholine to be removed from the synaptic cleft. Okay, so how does this work? Well, basically, attached to the postsynaptic neuron, you have acetylcholinesterase enzymes which break down acetylcholine. Now for short, acetylcholinesterase enzymes are abbreviated to ACH, and I should have said ACH is short for acetylcholine, okay, and then E for esterase, so acetylcholinesterase. Okay, now these enzymes are going to be attached in tetramers to the postsynaptic membrane here. So let me go into this in a little bit more detail. Let's say we have drawn the postsynaptic membrane out here, and I'll colour this in purple. Okay, and this side is the extracellular fluid, and this side is the cytoplasm. Okay, right, so the acetylcholinesterase enzymes in synapses like this, and I want to stress these are synapses in the brain, they're neural synapses. Okay, it's different at the neuromuscular junction. Okay, the way acetylcholinesterase is organised at the neuromuscular junction is very different. Okay, we are looking at how it's organised at a neural synapse like this. Okay, right, so on the postsynaptic neuron, you have a protein, a single membrane spanning a single membrane spanning protein here, with its amino terminus extracellularly and its carboxy terminus intracellularly. And this protein is known as the primer protein. Okay, and the primer protein stands for the protein-rich membrane anchor. Okay, so let's put this in. So P is for protein. Okay, the RI is then for rich, and then the MA is for membrane, that's the M, and then the A is for anchor. Okay, and 4 acetylcholinesterase enzymes are going to end up attached to the amino terminus of this primer protein here. So these blobs that I'm showing here, these are 4 acetylcholinesterase enzymes. So one here in green, another one here, another one here, and another one here. Okay, right, so that's how you organise acetylcholinesterase enzymes into tetramers attached to this membrane-spanning protein on the postsynaptic neuron. So if I put this in here, now onto this picture, here is the primer protein, and now we're going to have the 4 acetylcholinesterase enzymes attached over here to the primer protein. Okay, so I think actually I'll colour the primer protein in blue. There we go. Right, and of course you're going to have loads of these things in the postsynaptic membrane here. And these acetylcholinesterase enzymes are then going to break down the acetylcholine into acetic acid and also choline. So if I get back the picture of acetylcholine, wherever it is, okay, it's back here. Okay, here is the picture of acetylcholine. These acetylcholinesterases are going to hydrolyze this ester link between the acetic acid here and the choline here. So they're going to bring in water, they're going to attach the alcohol group of the water molecule onto the carboxylic acid carbon. Okay, So effectively you're going to split water here, you're going to split this bond here, you're going to stick this alcohol group here onto that carbon and the hydrogen onto that oxygen, and you will create the alcohol choline and the acetic acid molecule back again. Okay, so the two products then of this cleavage are going to be acetic acid, okay, plus choline. Okay, now what will then happen is these products of this hydrolysis reaction will be reabsorbed by the presynaptic neuron, and then they'll be refused together to make acetylcholine in the axon terminal, which will then be stored in synaptic vesicles until it's ready to be released. Okay, now the idea is that if in Alzheimer's disease we've had loss of these cholinergic neurons, okay, and that's resulting now in too little cholinergic neurotransmission in the cerebral cortex, okay, so too little acetylcholine is being released onto the cerebral cortex neurons. If we were to block the acetylcholinesterase enzymes, then the acetylcholine wouldn't be degraded anymore by the acetylcholinesterase enzymes, so you'd now get more acetylcholine remaining in the synaptic cleft. Okay, and therefore you'd get more cholinergic neurotransmission. This would up cholinergic neurotransmission, and hopefully it would return it to a level similar to what it was before the neurons had degenerated. Okay, so the idea is that we can increase cholinergic neurotransmission by blocking these enzymes from degenerating the acetylcholine. Now, of course, in reality, we're not going to be able to block every single enzyme, and therefore eventually it will still be broken down, okay, but it will certainly slow down the breakdown of the acetylcholine. Okay, right, so this is what the acetylcholinesterase inhibitors are going to do. They're going to block these enzymes and thereby potentiate the acetylcholine signal, okay, because the acetylcholine will remain there for longer, and therefore hopefully you can raise cholinergic neurotransmission back to a level that it was prior to the degeneration. Okay, right, so let me give you some examples of acetylcholinesterase inhibitors which are used in Alzheimer's disease. So an old one was tacrine. Tacrine isn't really used anymore because of its severe side effects. Okay, some nicer and newer ones that have less side effects are donezepil, okay, and then also rivastigmine, okay, and then finally galantamine. Okay, so tacrine, donezepil, rivastigmine, and then galantamine are all examples of acetylcholinesterase inhibitors which have been used to treat Alzheimer's disease. Donezepil, rivastigmine, and galantamine are still used. Tacrine is very, very rarely, I imagine, used because of its horrendous peripheral side effects. It's very liver toxic. Okay, right. And these are used in mild to moderate Alzheimer's disease, so they can give some sort of cognitive boost to people who are suffering from the early stages of Alzheimer's disease, where you've only got loss of cognition really because these basal forebrain nuclei have degenerated, rather than because the cerebral cortex itself is starting to degenerate. In the later stages of Alzheimer's disease, you know, raising cholinergic neurotransmission in the cortex isn't going to do much at all when the cortex itself is actually degenerated. Okay. So, the other drug then that is used in Alzheimer's disease is Mementine. Okay, now Mementine is actually going to have an effect on the neurodegeneration itself. It's going to slow down the neurodegeneration. However, it is not going to stop the formation of the amyloid plaques or the neurofibrillary tangles. It is going to reduce the ability of those things to actually kill the neurons. So it's going to slow the neurodegenerative process. Okay, right. So, before I begin this discussion of how Mementine works, we need to know something about what excitotoxicity is. Okay, so the concept of excitotoxicity is that if you take, let's say, a petri dish of neurons, okay, so here we've got a petri dish on which we've grown some neurons, okay, and let's say you put them in a medium that contains a little concentration of glutamate, okay, so you put them in a medium that contains not even a big concentration of glutamate, just a chronic low concentration of glutamate. all the neurons die. Okay, that is the phenomenon of excitotoxicity, that exposing neurons to chronic glutamate causes them to die. Okay, now why is this? Okay, it is because glutamate opens certain receptors on the neurons, which we're going to come across a lot in a moment, okay, known as NMDA receptors, which are receptors for glutamate. They're named NMDA receptors rather than glutamate receptors because they are activated by this exogenous agonist NMDA, N-methyl diaspartate. Okay, but they are really glutamate receptors. There are many different types of glutamate receptors. There's the NMDA receptors, the AMPA receptors, the kainate receptors. Okay, but they're all glutamate receptors, and they're all ligand-gated ion channels for glutamate. Okay, so glutamate is going to activate a type of receptor known as an NMDA receptor. Okay, these NMDA receptors are ligand-gated ion channels in the cell membrane. Okay, so if I draw the cell membrane here, here is our simple picture of our NMDA receptor. It's now open, and it's now going to allow ions to move through it. Okay, and the problem with NMDA receptors is that they allow calcium ions to move through them. Okay, the other glutamate receptors, the AMPA receptors and the kainate receptors, don't let calcium move through them. Okay, at least most of the AMPA receptors don't let calcium through them. But the NMDA receptors let a huge amount of calcium through them. Okay, now calcium is a lot higher in the extracellular fluid than in the cytoplasm. It's around 1.5 millimolar in the extracellular fluid, whereas it's kept at around 100 nanomolar inside the cytoplasm of cells. That is a 15,000-fold gradient across the cell membrane. Okay, now it's kept so low because high concentrations of calcium, I'm afraid to say, are extremely toxic to cells. Okay, the reason that high concentrations of calcium is extremely toxic to cells is still a little bit controversial, but it's certainly known that high concentrations of calcium in the cytoplasm of cells is just toxic. It kills cells. Okay, so if you're going to put cells in chronic glutamate, this glutamate is going to keep these NMDA receptors open chronically, okay, and you're going to be allowing calcium to move into the cytoplasm of the cell chronically, and therefore calcium level in the cytoplasm of the cell is going to get higher and higher and higher. Okay, and this high calcium concentration inside the cytoplasm of the cell is what's going to kill the neuron. Okay, so that's the phenomenon of excitotoxicity. Now, why is it called excitotoxicity? Well, glutamate, remember, is the main excitatory neurotransmitter in the brain. Okay, hence why it's called excitotoxicity. It's toxicity by overexcitation, effectively. Okay, so in the brain, the reason this doesn't happen is that you expose cells to glutamate for a tiny little period. You release a little bit of glutamate on. In a normal glutamatergic synapse, you'll have an axon terminal here, okay? Then you'll have your dendritic spine here. The axon terminal will release the glutamate onto the axon terminal, onto the dendritic spine here, okay? It will open up the receptors for glutamate on the postsynaptic membrane, which will include AMPA receptors, NMDA receptors usually, okay? So both AMPA and NMDA receptors, I'll put these on here. So let's have our AMPA receptors here. We'll come back to this picture later on. And then also the NMDA receptors here. Okay, and yes, the NMDA receptors will allow in a little bit of calcium, but it's only briefly. It only gives a little spike of calcium, okay, which is important for signaling processes, okay, and then the glutamate will be gone. The glutamate will be being removed by the presynaptic neuron, okay, and you don't have this chronic opening of the NMDA receptors, because once the glutamate is gone, the NMDA receptors will then shut, okay, and for short, the NMDA receptors are often abbreviated as NMDARs, like that, and this one here is an AMPA receptor, which again is a type of glutamate receptor. Okay, right, so usually you don't get enough calcium in to actually kill the neuron, but if they're open all the time because the glutamate is permanently present, that's going to cause calcium to go into the cell, and then the calcium rises to dangerous levels and causes the death of the cell. Okay, so that's the phenomenon of excitotoxicity. Okay, so Mementine is going to stop excitotoxicity by blocking NMDA receptors. It's going to stop calcium coming through NMDA receptors. Okay, it's an open channel blocker. Literally, it tries to go through the channel, gets stuck. Okay, binds in the middle of the channel, and then blocks calcium from being able to come through. Okay, so if you do have chronically high glutamate, okay, if you then put Mementine onto these cells, even though the glutamate was present and was opening the NMDA receptors and keeping them open chronically, okay, the Mementine would go in, block the pores, and then would stop the calcium going up within the cells, and therefore would block the excitotoxicity. Okay. Now, Mementine is known to have a modest effect in Alzheimer's disease at preventing neurodegeneration. It prevents the progression of the disease. It stops the neurons degenerating as rapidly. Okay, so what does this suggest? This suggests that maybe excitotoxicity is occurring in Alzheimer's disease. Maybe the amyloid plaques or the high levels of amyloid beta peptides or the neurofibrillary tangles, one of those things maybe causes problems with glutamate handling. Okay, maybe the neurons start releasing far too much glutamate. They leave glutamate chronically within the extracellular fluid. Okay, so at a high level in the extracellular fluid chronically, okay, and then that high level of glutamate in the extracellular fluid kills the neurons. So maybe Alzheimer's disease, actually, these amyloid plaques and these neurofibrillary tangles, they are just leading on to excitotoxicity, and then it's excitotoxicity that is killing the neurons. Okay, right, so that's the suggestion of how Mementine maybe works to slow down the neurodegeneration in Alzheimer's disease. Okay, now, I want to discuss this in a lot more detail. I want to firstly discuss the NMDA receptors in a lot more detail, because they are very, very complicated ligand-gated ion channels. Firstly, there's the complicated thing about the co-ligand glycine. Then there's the complicated thing about them requiring voltage activation as well. And then there's the complicated thing about why giving this drug doesn't actually cause major problems. Why doesn't it block all NMDA neurotransmission and cause absolutely major problems? Okay, and I want to discuss why Mementine can be given without it causing major side effects. Okay, because it is a very well tolerated drug. Okay, and it does produce a modest improvement in the neurodegenerative process of Alzheimer's disease. Okay, so in the next video what we'll do is we'll firstly discuss the structure of the NMDA receptor and how it works, and then we'll look at what Mementine does in more detail, and we'll look at why Mementine doesn't block normal physiological neurotransmission, but then does block this chronic excitotoxic stimulation of NMDA receptors.